doesn't seem to be working. Okay, that's right. All right. And the pointer goes like that. Good. All right. Thank you for coming to my talk. So today I will talk about finding new non-coding RNA genes with a new RNA-Seq method called TGIRT-Seq. Before we get into that, I have to talk to you a bit about RNAs and RNA structures, though. So as you know, there's a wide variety of RNA types and structures, and these structures are, of course, essential to these RNAs' function. And there are different types of structures, and some are more energetically stable than others. So let's say these transfer RNAs and HACase no RNAs have much more RNA binding that are stable than these CD-SNO RNA, for example. I'm telling you this because this actually has some implication in RNA sequencing. So as you may know, when you do RNA sequencing, you have to build a library, and to do that, typically you have to do a reverse transcription step. And to do this, you need a retroviral RT or an E. coli RT that reverse transcribes your RNA. And to do that, it needs a single-stranded RNA substrate in order to do the reverse transcription. It's good, eh? All right. But the problem comes when you have a highly structured RNA, which is hardly ever in a single-stranded form. Because of that, the RT just cannot do its work. It just cannot start the reverse transcription. And because of that, we have a big problem to sequence the highly structured RNA. And this is a problem to us in our group because we're very interested in some types of highly structured RNAs, such as HACase no RNAs, for example. And one solution was brought to that problem by the Alan Lembovitz group at the University of Texas, who brought a new enzyme to do the job. And its name is thermostable group 2 intron reverse transcriptase, or TGIRT for short. And its main advantage is that it's working at high temperature, which means that if you raise the temperature, your highly structured RNA becomes denatured. And that makes it so that the TGIRT is able to do its job. So we can basically, just like that, reverse transcribe highly structured RNA and therefore detect them in RNA-Seq. So this is a publication we made this year. So on the left, it's the classical retroviral RT RNA-Seq, and on the right, it's the TGIRT-Seq. If I may just bring your attention to the green slice, which corresponds to transfer RNA. On the left, you can barely detect any, less than 0.01%. And on the right, you can detect as much as 50% of transcripts that are transfer RNA. This is enormous, but this makes sense because you expect tRNAs to be very abundant in cells. So we think this is a much more true pictures of the transcriptome. So this was very cool for us because, like I said, in our group, we were very interested in highly structured RNAs, such as SNO RNAs, for example. But I began at this point to realize that I was detecting more than just these SNO RNAs and tRNAs. I was actually beginning to detect RNAs that were not even annotated. So this is such an example. If you can see here, this is a read alignment graph. And you can see these reads that are aligning here in the middle of an intron of this protein coding gene. And they have no non-coding RNA gene annotation on this specific spot. So when I noticed that, I decided to go in and investigate that further. I decided to identify all these read clusters with Blockbuster. So it's a tool that finds out the overlapping reads. It detects clusters of overlapping reads. And so I did that on a TGIRT-Seq data sets that were ribodepleted and non-fragmented. And this allowed me to find 1,212 clusters that had at least 100 reads and that were at most 500 nucleotides long. So I kept focused on small non-coding RNAs. All right. So I wanted to compare these clusters here with the coordinates with annotations. And to do that, I used the RNA Central annotation, which is very inclusive. It takes the annotation of 27 different non-coding RNA databases. And so I found out that there was as much as 266 of my read cluster that were not even annotated, so, which is enormous. But in that, I had to discard quite a few for many reasons, including too few uniquely mapped reads, length being too short, they were detected in just one replicate, they were antisense artifact, and there was one that was actually just a retained intron. So that left me with 146 potential new non-coding RNA. And I validated by looking if I could detect them in other data sets, TGIRT-Seq data sets. And for all these 146 genes here, I could detect them in at least one other data set of TGIRT-Seq that were in other tissues, cell lines, or even cancer. So that gave me good confidence that these RNA were not just artifacts of SCOV3-IP1 cell line, which was the cell line I used to make my data sets, and were actually true genes. That left me with 146 potential new non-coding RNA genes. So at this point I asked myself, how should I categorize these new genes? I began by looking for sequence similarity, so I decided to find if there were any homologs of my new non-coding RNA genes in RNA Central. I wanted to look if they had any structural similarity with known RNA classes. So I decided to try to find if there were any common structures between my new genes and SNO-RNAs or tRNAs using turbophobe 2 that was presented a bit sooner. I did function prediction as well, so I ran the SNO scan, SNO GPS, SNO report for SNO-RNA prediction and tRNA scan for tRNA prediction. And I've looked for association with repeated elements, namely tRNA repeats that are not necessarily annotated as tRNA genes, and ALU elements, which are very common in primates, and recent evidence show that these ALU elements can actually form HACA SNO-RNAs. So this may be indicative that these genes might be HACA SNO-RNA. So anyway, I compiled all those evidences, and I ended up deciding the most likely biotype for all of these clusters. And that gave me this big pie chart here, showing that most of these new genes are tRNA fragments, SNO-RNAs, and about more than a third are actually unknown, so they do not look like anything, and I'm a bit clueless as to what they truly are right now. But I may just talk to you a bit more about them, if we can talk about the expression. So this is just to show you the expression of the different new non-coding RNA genes. So most have a very moderate expression, around 10 counts per million, but some of them are very highly expressed. So if you see these tRNAs here, have more than 9,000 counts per million, and even this unknown here, which has about 5,000 counts per million, so which is enormous for RNA. Nobody knows what it is, and yeah. And for the conservation, most of these non-coding RNA are very lowly expressed, if you can see from the FASCOM score here. But the tRNAs and tRNA fragments are very highly expressed, and you can see a few exceptions here that are, highly conserved. A few exceptions, this unknown here, which is also highly conserved. So anyway, what is the biological relevance of these new non-coding RNAs? So I told you that many of them look like tRNAs. This is just an example of a result to show you that they probably function as true tRNAs. So these 4 blue boxes here are actually 4 new tRNAs, and these red boxes here are annotated tRNAs in the RNA Central. So you can see these new tRNAs are very proximal to these annotated tRNAs, and this is most probably a tRNA cluster. And if you can see here, these 3 tracks are actually polymerase III proteins chip data set, which shows us that this whole cluster of tRNA is actively transcribed by polymerase III, which is expected for tRNAs. And if you can see, this is just one example, the cluster 6. It folds very much like a tRNA, it has an anticodon, so we expect that it probably functions as a tRNA. But this is not necessarily true. I'm not so sure of that, because many of these tRNAs actually have many flaws in their sequence and structure that makes it so that they are not predicted as being tRNAs by tRNA scan. So mainly smart or short stems, mismatches, or lack of CCA3' tails, things like that. That may indicate that actually these new tRNAs may actually have non-canonical function. So we just do not know yet. All right, this is an example of SNO RNAs now. So just to tell you, there's about half of these 26 SNO RNAs that are predicted as having a ribosomal RNA target. So we expect SNO RNAs to have ribosomal RNA target. This is one example here of one of these. It's included in the intron of this protein coding gene. It can fold like an HACA SNO RNA. It has the H box and ACA box that is expected of an HACA SNO RNA. And you can see its guide sequence here to bind to the 28S ribosomal RNA to modify this specific nucleotide. But still, there's more than half of these SNO RNAs that have no ribosomal targets, so it might be indicative that these SNO RNAs as well might have non-canonical functions. And this is a very hot field for the SNO RNAs, by the way. There's a lot of non-canonical functions that are talked about recently. And finally, just one last result I could show you with these new non-coding RNA genes. So these two graphs on the left and the right are actually the same, but just with different colors. And this is just to show you the difference in expression between benign ovarian cancer and aggressive ovarian cancer. And I just wanted to show you here that many of these new genes have a very significantly differential expression between this aggressive and benign cancer. Many of these new non-coding RNA genes are much higher expressed in aggressive cancer, namely many of these SNO RNA HACA genes, many of these tRNA genes, and this fragment here that is actually a fragment of an HACA SNO RNA by sequence comparison, and actually has about 10,000 counts per million in aggressive cancer, and has barely just 10 count per million in the benign cancer, which means that there's a 1,000-time increase in expression in aggressive cancer. So, which is very interesting and tells us that these new genes might have an involvement in cancer cell proliferation and might actually be new biomarkers. But yeah, as of yet, we do not know the true function of these new non-coding RNAs, but we have these few clues here. So, just in conclusion, let's say there would be 146 new genes that may be added to annotations, including many tRNAs and SNO RNAs. They were not annotated before because they were hard to detect because they were highly structured. Many of them were atypical, so they would not be predicted as being tRNAs or SNO RNAs, let's say. And many of them have low conservation, which does not help. So yeah, some have potential involvement in cancer cell proliferation. And you should, of course, reduce your RNA structure bias for better transcriptome evaluation by using TGIRT. For future direction, I would like to sequence more tissues and cell lines with this new RNA-Seq method. I would like to investigate the interactions of my new non-coding RNA genes with other RNAs or proteins. I would like to look at their expression after the depletion of key proteins, after the depletion of key proteins, such as proteins that are involved in SNO RNA complexes. I would like to make the depletion or the deletion of some of these new genes to potentially investigate their function. And I would, of course, like to investigate the unknowns, so these 50 new non-coding RNA genes that I have no clue what they are, because they do not look like anything and they do not fold like anything I know of. So yeah, with that I would like to acknowledge my two labs, especially Olivier Boivin, which was an internship in our lab, which helped me quite a lot with this project. Also the RNA-Mix platform, which did a lot of work, wet lab work, and the ISMB for Travels with Fellowship that helped me come here, and you for your attention, of course. And I will take your questions if you have any. Yeah, go ahead. Thanks.
So very cool work.
Thanks.
I guess the first question for you and the previous speaker would be, following last night, is how many genes do we actually have? Right? And then, so the other thing is, I'm guessing you're thinking about this, but one possibility is these are, leftovers or things that are dying out because it's just, they're it's just similar to pseudogenes or things that are non-functional anymore.
Totally.
But if you have cancer, things start being activated all over, then things shoot up.
Yeah, exactly. But I looked at their expression in tissues, not just cancer cell lines. So that gave me a good confidence that they, if I can find it, yeah, right there. So I look in prostate, no, I can't find it. All right, it's this button. I looked in prostate, breast, and ovary to see if they were expressed. Many of them were expressed. But of course, it's entirely possible that many of them can be cancer-specific artifacts, you could say. And many of them could perhaps have no functions at all, and they may be just left over from evolution, copies that happened and that were degenerated through time, through evolutionary time. But yeah, there's just no way to tell right now. A bit too soon in this project.
But for the TGIRT, so can you go back to regular RNA-Seq and still find hints of those now that you should be looking, and then maybe you can look across tissues in GTEx or...
Yeah, actually, I haven't looked in too many data sets, but I've looked in the SCOV3IP1 data set that were not done by our TGIRT-Seq, that were done with classical RT. And I actually could detect quite a few of them, more than half of them I could detect them, but with very low expression level. And I took these data sets because they were like full lane sequencing, like 250 million reads in one data set. So they had a very high depth of sequencing. So that's why I could detect them. But in lower, more conventional sequencing depth, I could just not detect them. So yeah, the thing is you just need a more sequencing depth to detect them with classical RNA-Seq.
So speaking of classical RNA-Seq, when you look at the coverage plots in a genome browser, we're used to seeing that characteristic kind of peak and valley pattern, which, there's some indication that's affected by GC content, and it's also been thrown out that it could be secondary structure. Do you see that goes away and you see a lot more uniform coverage with your TGIRT-Seq?
Totally. And most interestingly, I've done this specific data set as being non-fragmented as well. And the reason why we use this data set is that we see these accumulation on annotated gene much more clearly. There's no holes or bumps or whatever. It's just a big square like that shows us the beginning, a clear beginning and a clear end of each transcript. Although this is not perfect, I've noticed that in some specific RNA structures like G quadruplex that are very stable structure, I see drops in the accumulation of read on these specific G4, let's say. But mostly it's just flat like that, and it's very well defined, so it's very cool for that.
But that holds true for also just the known genes, not the novel ones, like if you were looking at
exactly.
Okay.
Exactly.
So you can use this to detect G quadruplexes?
Perhaps, yeah. This is one idea we had. We haven't investigated that yet, but yeah. We have to talk about that with Jean-Michel. We'll talk a bit later. Yes?
I have two questions. So, TGIRT, you're using manganese as a buffer for RT, right?
Oh, that I do not know.
If you're not using manganese just for normal RT, I bet that sort of gives you the same result, right? It's not the temperature that I think is unfolding just one of the curious, right?
I'm looking at my PI right now asking for help because I'm not too sure. I'm not doing the wet lab work.
The other question is maybe a TGIRT is, it's more sensitive to RNA modification from that. The TGIRT is typically very modified.
I do not know if it's less or more. Okay, yeah, sorry. I don't know if it's more or less, but I know it is still sensible to some RNA modification because there was this one paper that showed that he would use demethylase with TGIRT-Seq and they would have an even better detection of tRNAs. So it's still sensitive to modification.
Could you comment on any specific modifications which are where you're seeing a drop?
So I haven't noticed them, but I've been told that G1 methylation, A1 methylation, and C3 methylation do cause a decrease in detection. They actually block the reverse transcriptase sometimes.
Has it been shown before?
Yeah, it's been shown in
It
this.
drops off, it does. Okay.
I couldn't give you the title of the paper, but if you come to my poster, A21, I could tell you. Anyway, so thank you very much.